Okay, so we're actually briefly taking a step back to talk about more short reads, germline processing. Now, specifically, we're going to talk about the DRAGEN-GATK pipeline we have here, because it's got an interesting history and it's actually relevant to a couple questions. Like just this morning, a lady asked me, well, what can we do? Like, remember the region we had in the first demonstration that had like T, and it was really hard to kind of align to it and work with it? The question was, well, are there options? Believe it or not, I'm going to talk about exactly that here. That's one of the updates we made over a base GTK, specifically in DRAGEN-GTK. I'll spend some time talking about what some of the specific error models are and how they help with things like this. So, in terms of history, well, there's a lot that happened after GTK 3. Okay, so we put GTK 3 out there, our best practices pipeline, and it was awesome. At the time, it became an industry standard. Now, the thing is, several companies started with GTK 3 and said, can we do better? Can we optimize this? There is Parabricks optimized GTK 3 using GPUs, and they're required by NVIDIA. There is Synteon optimized it by rewriting it in very fast C. There was Edico, a company that optimized it using C and FPGAs, so specialized hardware there. They were later bought up by Illumina, and that's what became Dragon. Now, later, Illumina reached out to us for cooperation, and we said, sure, of course, you know. And thus, the partnership between that group, Edico, acquired by Illumina, and the Broad is what you're seeing here when we refer to DRAGEN-GTK. That is a partnership between our organizations. They kind of branched off from us after GTK 3, and we came back together. Now, the thing is, it wasn't just about making our pipeline faster, though. There were actual scientific improvements that their team made over time, independently from ours. Now, we made improvements too, but both teams had made improvements. So we figured, okay, we came together. Our engineers directly met up. We had a lot of cooperation. We directly put over algorithms into GTK that came from their work over there. Some vice versa. I'll mention some at the end that independent advances we've made that they haven't, but there's a lot of cooperation there. And the goal with that, because Dragon still have their hardware optimized software, they still have it, but with DRAGEN-GTK, the idea that you'd still be able to run it somewhere else, not on this super expensive hardware, and you'd still get results that are directly comparable. Now, I'll talk about what it means again to be comparable. Now, Dragon and DRAGEN-GTK now do produce very close results, and they actually outperform classic GTK. Now, what is this curve? I'll take a moment to talk about that a bit, the ROC curve. When I was talking about the filtering model there, I mentioned how when you're filtering sometimes that you end up with the whole purpose is to kind of reduce the number of variants that you incorrectly call. And in a perfect world, right, you also wouldn't toss out any new variants in the process. But that's not how it always works. When you look at performance here, there's always going to be a trade-off. This is it's called a ROC curve. So you see along the bottom, you have your false positive rate, and along the top, you have a sensitivity, which is how many of the actual variants you're catching, reflected by your false negative rate. And what you want to be is in the top left corner, because in a perfect world, in that corner there, you're not getting any more false positives while you're getting rid of all your false negatives. That would be a perfect world that goes right up in the corner there. So any of the curves that go better into that top left corner are doing better. It means the trade-off isn't quite as steep. Because as they go down here, it means that in order to reduce your false positives, you're having to take on a lot more false negatives. That's what a curve that's down here is. Up here, it means you're able to do filtering better without introducing more errors, basically. And you can see that the old classic GTK, it did okay. Dragon did better, and DRAGEN-GTK actually does even better. That's why we bother with it, because ultimately, we're trying to get better results. Now, also something important to mention, the changes I'm going to talk to you about today are actually currently baked into GTK 4. They're in there. We have a standard pipeline now. I wasn't in this room on the first day when they asked a question about pipelining and how many of you run pipelines and are familiar with them, but we do have a GATK best practices pipeline. And in that window, we've already configured it to do everything we're going to talk about here today. Even if you use the tools directly, I'll mention at the end some how if you're not using our pipeline, you can just kind of run the tool and still get some of these optimizations in there. All right. But again, everything we taught so far is valid, everything you can do, but this is a slightly modified pipeline in the specific case where we have to bring our new GATK in line a bit more with what Dragon does. And like I said, the best practices version that's up there in Terra already does these things. So as I mentioned, we want to bring our pipelines in line. What did that involve? What are some of the changes we had there? Well, here's a standard pipeline as you're used to seeing it. This is actually our single sample pipeline though. The other one we mentioned had like joint calling in there, right? And you see here, it's not. This is a single sample pipeline. You call variants per sample, you spit out a GVCF, you have your raw snips, you use the CNN, the convolutional neural network. Eric was just talking about it. That's a single sample one, right? So you don't see any joint calling in here or anything else. Sometimes you do want single samples. A lot of the advances that Dragon made were really focused on that use case, specifically making better single sample variant calls. Now, that still affects joint calling because ultimately having a better called sample is better input when you're going to joint call them together. But you'll find that a lot of the actual changes are optimized to try to do that. The goal was to do the best possible single sample calling on a human genome. That was the goal. And the Dragon pipeline is, it's similar-ish. You still see alignment, but they have their own aligner. You still see a bunch of stuff we talked about before. Marking duplicates, GTK does it. Detect active regions, that's a GTK thing. Assembly, that's fine. A hidden Markov model, again, we're aware of that. They've got this other thing here. They train a PCR model, that's new. So they feed some stuff into the hidden Markov model. I'll talk about that. And then they go out to the genotyper. So it's analogous in many ways to what we did. So in terms of some of the final changes we had to make, it's worth noting that it's used a different aligner. There's now they released their aligner, and open source is called DragMap. So the DRAGEN-GATK pipeline uses DragMap. So there's a little difference there on the aligner. You'll note that there is no BQSR step. That step is missing. I'll tell you why later. There's another step we introduced that actually serves the same purpose and does it just a bit better. So on the Dragon-GATK side, you won't be running BQSR. That's another change we made over our baseline pipeline. Also, after you've done all the standard stuff you're used to, alignment, variant discovery, and all that, there's a new era model. There's something called the SCR model. I'll explain in detail what that means and how it works. That's introduced there, and we have to learn it per sample. And at the end, when we're calling variants, they still call haplotype caller. Like haplotype caller is still in there. It's just a slightly different argument to it. So again, we tweaked the pipeline a little bit. And at the end, I'll just point out that you can still do the neural network. In principle, it takes a little wrangling to make the results exactly line up, so you can run them through that again. It's a little bit of wrangling you have to do there, but it can be done in theory though. And again, these aren't changes you'd have to make yourself. I really want to stress that. If you use the pipeline we have available, I'm describing what it does, not things you'd have to change, okay? These are changes we made in that particular pipeline. So let's talk a little bit about what the improvements were and what they gave us. So DRAGEN-GATK changes the core GATK haplotype caller a bit. There are modifications there. You remember the haplotype caller. We've talked about that a bit. I think you've seen a version of the slide a few times. That's the part we're changing down there, okay? Yeah, all the changes I talked today are going to fall within the red box there, either the left part or the right part. And there are 3 specific big algorithmic changes there. And I'll point out, especially the first 2 are another example of how sometimes you can bring in a little more data to your calls and do better. There are different ways of doing that here that it can improve the quality, but I think they're really clever, actually, and they help us out. I'll bring in that Dragon explicitly modeled a couple different error modes of the sequencer, because we know sequencers have errors, right? But it's one thing to say, broadly speaking, they have errors. It's another to say, can we actually model those errors in a meaningful way and account for them? And they did a good job modeling certain classes of those errors. The SCR model I mentioned up there, again, I'll go over in much detail in the very next slide what that is. It's modeling regions like I mentioned before, where you have a lot of repeats that are hard to map to. It's something you do in pre-processing, and it modifies the hidden Markov model. It directly feeds in, modify the hidden Markov model a little bit. The other ones there, base quality dropout and the foreign read detection, they come in the next stage in the genotyper. And again, a lot of it is fixing. We make assumptions from the first pass, like the independence of reads, and we know that there are things that happen in sequencer that makes them not actually independent. And I'll go into more detail again about how exactly the BQSR handles that and what the effects we get from that. Let's go over the STR. So an STR is a short tandem repeat. Remember we saw this? The exercise we did yesterday, we saw that in NA12878, we had that area, T. Remember that? And it was hard to align, and it wasn't feeling confident making calls. It's not just TTT. Sometimes it can have a longer period that repeats, like the top one, TC. That's also one of those types of regions. And also it can be a longer motif like TTG, and you can characterize them. Like how long is the repeating segment and how long does it go for? These are all over your genome. By the way, they're everywhere in there. And you imagine that gets a little bad when you're trying to sequence, right? But the thing is, it's not just like the aligner. Turns out the PCR also struggles on these regions. It has a tendency to insert or delete from these regions as well. So the errors aren't just in alignment, they're also like systematic errors that occur just in sequencing these regions. Furthermore, I'll point out these regions, the reason we have them is because they're hard biologically. Your cells make errors in these regions, okay? Like these are just difficult regions to work with. But we don't have to stop there. We can do a little better beyond just saying they are difficult. So what do we do with that? Well, there's this new tool called Calibrate Drag Stir Model. It's another tool we can run there. And what does it do? Well, it looks at the reference genome, human, right? And it scans through and says, well, let me find all of these regions across your genome. It's a pre-processing step. Just a further reference, let me characterize the entire genome and calculate them. There's a tool that does it, by the way. It's published somewhere. So you don't have to do this. But it goes in and says, all over your genome, here are these regions, right? Identifies them first. After the normal pre-processing steps and everything, we go by type, like for instance, period 3, length 5, right? And we go and we count all the errors where they are in the genome, and we try to calculate the baseline error rate. Now, there are a couple assumptions we have to do here. The first is that, realistically, each individual in here doesn't actually have all that many errors in their genome. So we don't really know, right, when we see a change in one of these errors, is it a PCR error, or did that person just have an indel in there? We don't know, but we're starting from an assumption that's almost right. And that assumption is that if one of these regions is different in your read, let's assume for a moment it's an error. Let's assume that. That's not true, but it's actually going to be close to true. And what that allows us to do is go in and characterize if you see a particular type of STR region, like, I don't know, period 3, length 5 or something, we get to establish a baseline error rate for that. Now, why does that matter? Because PCR errors can sometimes be a lot, but they can also vary sample to sample sometimes. It's not like you have an established baseline PCR error rate. Oh, maybe it's a lot in this particular sample, maybe it's not. So going through every sample as a pre-processing stage and saying, what do we assume the error rate to be in this particular sample lets us estimate how bad was PCR here, you know? It's not perfect. Again, when we see an indel in these regions, it could be real, but most of these changes probably aren't going to be. And what that allows us to do is actually figure out a baseline error rate that we can work in as a prior assumption. So for instance, I'll go back to this one here. So remember when we were talking about using the hidden Markov model to go and estimate likelihood, and we were talking about using Bayes' rule? Well, now if we have, now as our haplotype caller is going through, if they can identify, hey, this particular read here is entering one of these regions, it's entering an STR region, right? A short tandem repeat, we can now adjust our Bayesian priors based upon the calculated error rate we made over the file and say, well, we know this file has a certain borderline, I don't know what the number would be, a certain error rate we calculate in this file. Let's use that as a prior. Let's assume for a second that we know there's just a baseline error rate here. And let's use that in context to really say how confident we are in this call. Like if we know there's a high error rate, for instance, in the file and we enter a region, we're going to lower the confidence a little in our calls from there. If we know it's a very low error rate, we'll keep very high confidence. So this affects the hidden Markov model at that level of computing likelihoods for a given read, because it allows us to give better priors. We give better priors to our math there, and specifically in these regions. Now, okay, some of the math is on the right. I'm not going to go into it. And the end result of that, we get much better indels, much better, which is to say that we used to have about 60% accuracy on indels. Indels are hard. What can I say? Right? We used to have about 60% accuracy. When we put this error model in there, it jumped to high 90s. Like it was a significant change just to be able to account for those errors and use them as priors in doing our Bayesian math. Like that was a huge win on indels. Just like I said, bringing better context into one call. So that's one of the three changes, the STR error model. Now, the next ones come in actually at the stage after that. So that would go into how we're computing our likelihoods there. Well, the BQD and FDR error types there, they allow us to characterize the likelihood of getting a wrong call, another wrong call. Like even if we saw, let's say, I don't know, AC, it gives us a model to say, well, how likely was that to be AA, for instance, and C was actually an error? Or how likely was it to be CC and A was an error? They realized this is a thing that happens sometimes. I'm going to go into, again, next slide, the details for what BQD and FDR are. Don't get me wrong. But these two come in at the stage afterwards now. As opposed to just evaluating the particular genotypes there, it actually tosses in a couple more states to compare against. It's still going to, try to pick the one with the highest likelihood, right? But now it explicitly says, let me actually evaluate it against error. So if you look at the top line there, it might just say, well, looking at the number at the top, maybe it thinks that's a het call. Maybe it thinks it's AC. That one has the highest likelihood. But when we explicitly modeled the BQD and FDR error rates, it might come out and say, well, now the most likely thing is that it was actually AA and that it saw a C, but it kind of hallucinated that C. And now that we can model the likelihood of that happening, we can be a little more refined and actually downgrade that to a hum call there and say, you know what, actually, we don't think that's right. Now, it changes it. Not a huge confidence in our call there, but enough to tip which variant we would call. It was enough to offer another boost, not as dramatic as the SDR error, but it added another boost to our accuracy. Now, what is BQD doing, right? Really, what BQD is doing is saying, like, look, so let's take a look at the calls here for a second, right? On the left, we see a reasonable, we see a reasonable SNP here, right? But we see these other calls here in green, right? The question is, are they good? Are they not? We only see them. Like we only see them on other reads. Like we don't see them along with any of the good ones on the blue. They're only on separate reads there, but maybe they're real, maybe they're not. But if you look at them in isolation, you might not know. But let's look at the rest of the read for a second there. Got a lot of errors at the end, doesn't it? And this one you can see, looking at isolation, you don't know, but there are a lot of other errors. Errors kind of, or potential errors, let's say. We don't know they're errors, but potential errors. They cluster a little, right? And that's because when you're running the sequencer, it doesn't actually give every error completely independently. Sometimes sequencer is running and, driving along, engine catches on fire and the wheel pops off. And it's just kind of wandering for a while after that. And the truth is it's going to skip around. It's going to make a lot of bad calls there. And it happens like that. Sometimes one read will just, the whole read's going to have a cluster of bad calls. We know it. It's how they work. So what that means is maybe we shouldn't look at every call in isolation. Maybe we should say, okay, but you know, the rest of that read has a lot of potential issues here. So maybe knowing that, we might want to downgrade our confidence just a little bit in this one. So take the neighboring reads on that particular one right, into account, right? Also allows us to better account for the error because we know that the machines don't, I mean, the error is linked. They're linked. So this helps us bring in that context to an individual call by also taking into account the other errors in that particular read. And yes, in doing so, I mean, it actually does, as I mentioned, penalize alleles that have lower quality ones, and or that specifically are surrounded by lower quality reads. And that when they mention base quality dropout, that's what BQD stands for. It means that really sometimes when the sequencer goes bad, the quality drops off for a little while there. You're going to have a bunch of bad reads, and you should take that into account, and it does. These slides you have, if you really want to go into the actual math down there, you can. The last one is our foreign read detection model, okay? Now, we already do kind of a version of this, to be honest. We try to detect for contamination and everything. The truth is they just gave us a slightly better version. You look in, and it looks at allele depth a lot. Why does allele depth matter, right? If you're, let's say you have a real region, let's say let's say it's het, you got an A and let's say a G there, right? You'd imagine when you do the sequencing and alignment that you should get roughly half and half. Maybe half of your alleles would be an A, maybe half of them would be a G, right? If that's the reality. But what happens when the balance is wildly off? And like you see maybe 20 of one and only 4 of the other. Like, okay, that's a hint something's going on. So it tries to calculate the not just the allele depth, but it also goes down into very low map Q reads where the map quality is low. We often just toss them and ignore them. This part goes down into them and says, okay, well, also, did I only see this read on really, I only see the particular variant or potential allele on really bad reads? Like, is it really prevalent in the bad ones and none of the other ones? It takes that into account too. Before we kind of ignore them, but it's actually easier for it to say, all right, again, if I only see like the G's there, for instance, if they only show up on really bad reads, it's likely maybe it's not even real. You know, like those bad reads that don't align, that might be bacterial contamination or something. If I'm only seeing it there, then maybe what's actually happening is that the one that appears to align is the fluke, you know? And again, it's one of those things that helps us model contamination better. We had a model for that. This one's a little better. These incremental changes really do add up. And in the end, it does produce for us some pretty knowledgeable, it's a pretty better result. The original, the Dragon over the original core GATK. One thing I'll point out is that there is a little bimodal quality distribution here. You see the one around 100, and it's another kind of peak there around 50. That's actually an artifact of some of the new models. I'm maybe opinionated about that, but I'll keep it to myself. When we're modeling hets being potentially downgraded to HOMs, because of that, it reduces likelihood a bit in hets, and it tends to cap quality of the het calls around 50. It's more conservative in general with the quality it gives. It is. You should know this, but remember, it's designed to do better single sample calling. So even if the quality scores it gives are in general a little more conservative, if you cut off low, I think around 10 often in practice, it doesn't matter. You have a lot more confidence in where you cut off. The other thing is because it's a little more conservative in how it actually assigns quality values, believe it or not, that value is actually a little better here. It's a better indicator of the true quality of the read before we often have to run it through VQSR or VETS or something like that before we're really confident in it. It's more conservative, but actually gives us a more reliable value. Why is that important? We're talking single sample calling, remember? That means we can't really train a giant machine learning algorithm on it. In a single sample case, we're really working with what we have within this sample. That's the idea. So in that case, it's okay if the overall quality is lower, but more reliable maybe. Now, these are built into the GATK tools, okay? In fact, if you really want to just run it from the command line, you can add -- dragon mode. And bam, all the things we mentioned there, it's going to turn them on. And it'll use them. They're already there in GATK. There are a couple other arguments, though, if you want to use that. When you run that, by the way, I believe it'll also spit out diagnostically. That's kind of a global flag there. There are other individual flags. You can turn each of the models on individually. Want to use an STR model? Cool. Like you can use them individually like that, or maybe you don't want it because your particular use case, you might know specific things about your use case, right? Maybe it's not human. There might be specific things that want you to use one error model and not the other. Oh, and the dragster params path, that's the argument you have to pass. And when you call calibrate dragster model, again, that one gives us we do a lot better on indels with that. If you call that, it produces the file. That's how you pass it in. You pass in the -- dragster params path, and you get to use the STR model then. Remember, the STR model, the inputs for that are calculated as a pre-processing step. But you can use these things. Again, I'll stress that they're in there. They're in our pipeline up there. If you look at our whole genome germline pipeline, these changes will be in there already, okay? If you're comfortable running the pipeline, you kind of get them already for free, I'd say. Now, also, why are those particular versions of Dragon targeted? It mentioned that supports Dragon 3.4.12 and 3.7.8 are coming. Well, that's because, honestly, those are the versions of Dragon that are targeted by large biobanks. I mean, I can tell you right now, the last major AOU call set was done in 3.4.12. The one we're doing right now, as we speak, is 3.7.8. So we're kind of catching up to the ones that are in use. And it's more important to match those and sink the labor in to match those versions of Dragon because they're the ones that are going to be used a lot. A lot of people want to align with these big call sets so they can compare their data against them in a meaningful way. So 3.7.8 isn't here yet, but 3.7.8 equivalency is coming. All right, now I say equivalency. It's worth asking what we define that. What does it mean to say we're kind of equivalent to it? Because, I mean, we're not exactly equivalent to it. I mean, Dragon GATK is just not byte identical to Dragon. It's hard. They're written in different software. Some of it's in hardware in Dragon. Like, they're not exactly equivalent. But the question is, what's good enough? Now, you have to ask the question because if you just randomly throw in samples that are processed in different ways, you introduce batch effects. Batch effects are errors that come from the fact that a particular way of sequencing, might introduce a certain bias in there. Mix them in together, you'll have what look like differences in the underlying data, but really all you're showing are differences in biases in your different pipelines. And different pipelines introduce different ones. So you want to be as consistent as possible, but you also want to be able to say, if at all possible, is there a way for me to mix and match pipelines while minimizing these effects? Is there a way? Well, that's what functional equivalence tries to answer. How can you really measure that? And the idea is that, well, there's a certain amount of change you're going to see due to, well, called the weather. You sequence something twice, you run it twice, things are going to change, and you're just going to get slightly different results. You're not going to get the exact same result twice, right? So we want to tease apart something. What fraction of the errors we're seeing and differences we're seeing, really differences, come down to the fact that we're using different toolkits to process the data versus the fact that they're just run twice. So we try to figure that out. So what we do is the technique is to take a sample, run it twice, process it twice, right? And for each of those samples, run it through the two pipelines you're comparing. In this case, Dragon and Dragon GATK, right? Run it through the two pipelines. And at that point, you have an interesting data set there. You can compare things like, well, I've sequenced it twice, but I've processed it the same way. So I've sequenced it twice, but I'm processing using GATK. How big is the difference? Again, same pipeline, just sequenced twice. You can actually calculate that. How big of a difference is there? And you can also ask, okay, how big is the difference just running it twice with Dragon? How big is that? And then you can ask the question, well, okay, fine. I know a baseline difference just from resequencing. I can estimate that. How big is the difference I get due to processing them differently now? How much does that introduce? Because that's going to introduce some difference. I mean, again, they're not byte equivalent, right? But now you have a metric. You can compare two things. You can say, how big was the difference just by, the weather when they were run, right? Two different pipelines versus the differences we introduce by using two different ways of computing. And the idea is that if we can get the error low enough between the two different pipelines, and we can say, well, the error that we introduce due to our tooling is less than the error you introduced just by sequencing them twice. At that point, we feel safe saying they are functionally equivalent. Now, what are the tools we use? Well, we look at concordance. We mentioned concordance a few times, right? The concordance tools, you measure false positives, you measure false negatives, you measure two positives, you calculate some statistics. I mentioned it earlier in call set evaluation. And also, I believe, it came up in at least one talk earlier. I believe Eric mentioned concordance as well at some point, but it comes up. We use concordance to calculate it, but we also look at the differences in quality there, and I'll mention why later. So, concordance works. We went over this before. You define one of the samples as truth, the other as evaluation sample, run VCF eval, calculate your concordance, and there you go. You have your number. I'm skipping this one, moved it earlier. So, what do you do at this point? You say, well, first, you try to look at the ROC curve here. You're like, do they line up? They should, ideally, they should pretty much line up here. You shouldn't see a big difference when you calculate concordance between them, right? So that's a really good example that you actually have it. You actually have a really good area of overlap there. Well, there's another one though. See, the problem is concordance isn't the end of the story by itself. What if you have two different pipelines, right? Two different pipelines. They actually agree on what variants are good and which variants are bad, but one pipeline has a bias. It systematically assigns a +10 to the quality score. Depending on where you do your cutoff, it still might agree on what's good or bad, but you can't really compare the two now. You can't put them together and compare them because suddenly one will look like it has a higher quality than the other. So we actually there's a different metric we have here where we look at the score differences and we evaluate for different cutoff points in quality. We look at different cutoff points and see how big is the difference there for the scores. And the long and short of it is that you want the green line to drop below the others here. And at that quality cutoff, you can say that at this point, they're equivalent not just in overall concordance, but also if you cut your threshold here, you'll see equivalent results too. Now, the functional equivalence pipeline is also available in Terra. Okay, I'll point out that the functional equivalence pipeline, we don't just use it to compare, for instance, DRAGEN-GATK to the original Dragon. We actually, on our pipeline there, you see us compared GATK3 to GATK4. You'll see that in there as well. And what we find there is that while the two are highly concordant, they are not functionally equivalent, for instance. So it should give an example. You can, in theory, compare any two kits, really, to see how close they are to each other. And it helps when you're trying to mix and match data. So again, you can go here. You can find, there's a link on the home page that would take you to the DRAGEN-GATK page. And that's where most of our articles, workspaces, videos, all the resources we have related to that, you can find them there. Okay? And one last thing I want to mention. I mentioned that, by the way, that there are different changes that the two independent groups actually made their own improvements. One of them that we made was Dragon doesn't actually model spaying deletions all that better. A spaying deletion is just a really, really long deletion there. It's hard to model them. We actually did a little better on our side. Eric can go into more detail on that. We did better on the GTK side and how we model spaying deletions. That's not turned on by default in DRAGEN-GTK, but it's an example of you can turn on in yours though. If you happen to have a sample with a lot of spaying deletion, that's an example of a change we made. They haven't quite incorporated back into Dragon yet, but it's something that you can turn on. Remember the ROC curves. When we actually look at running it with spaying deletions, we can do even better. Even better. It's a constant game of inches, squeezing yourself closer to the top left quadrant there. But there are constant changes as we work with them. I think that's actually about all I had to talk about here. Yeah, exactly. I don't need the other ones here. So, if you have any questions? It's kind of a lot I threw at you there. Hello.
Do you recommend to run STR error model only with PCR samples?
That's a good question. Should we run the STR error model only with PCR samples? Yeah, Eric says no. Gotcha. Okay, so okay, so our question was, should we run the STR error model only with the PCR samples? And the answer is no, we should run it all the time. And what Eric pointed out was PCR-free genomes still use PCR. They actually still do. Do we have any other questions? I don't know if we have any from the other room, so I'll give them a moment in case someone there has a question that they bring to us. If not, then I'm sure we can take our short break and move on. I, for the record, happen to think the stuff's really cool because my primary focus, like I said, is in joint calling, but the very nature of that is bringing additional information to bear on a particular call that's ambiguous. Anything we can do to get ourselves more reliability and more confidence in what we're doing. So there are lots of ways in which people have found ways to do that and bring more in context to bear. All right, any questions over there? All right then. Yes, we'll take our. Yep, we'll have our 10-minute break, and then we'll be back a little bit after that. And I believe we're doing the long reads module after that. Okay, see ya.
All right, we're going to start up again. I'll pass the microphone over to Emma in 1 second, but there was a good question I want to tell everyone. So the question is, if you are running on an Illumina machine and you have Dragon running on the machine, can you choose the version of Dragon running? And the answer is, for the most part, yes. But something's very important. The Illumina just came out with some new machines called the NovaSeq X's or the Nova X's. And they, for some reason, do not allow you to use older versions of Dragon on the Nova X's. And that's important because everyone in the world is using the older versions of Dragon. Okay, so the UK Biobank is all on the older version of the Dragon. The Genomics England, all on an older version of Dragon. All the Broad stuff, all of us and the NIH, all on the older version. And they just moved to the older version of Dragon, and they said they're not moving off of it. So if you are running on a NovaSeq X on a new machine, you'll actually have to reprocess your data. If you want to be consistent with Nomad and all of us and UK Biobank, if you don't, then it doesn't matter. Okay, so I'm happy to answer more questions afterwards, but for now I will pass this on to Emma, and we'll stick around till afterwards. You can ask lots and lots of questions when she's done.
Yeah, there should be plenty of time at the end for additional questions at the end of the day. Okay, so I'm going to talk to you a little bit about long read sequencing. So far we've been really focused on short read sequencing. So the leftmost image here is one of the Illumina sequencers that does short read sequencing, and that's really all we've covered so far. But there are these newer long read sequencing technologies that are starting to become more popular. So we're going to talk a little bit about some of the advantages of using those newer long read technologies. And there are kind of two main long read technologies that we'll discuss. So in the middle, there's a sequencer from Pacific Biosciences, or PacBio. And on the right, there are a couple of different sequencers from Oxford Nanopore, or ONT. So there's the Prometheon on top, which is a little bit larger and higher throughput. And there's the MinION at the bottom. It's a really tiny sequencer, which makes it extremely portable. You can actually do sequencing like in the field, but it is lower throughput because it is smaller. So why do we want our reads to be longer? One answer is that that enables more comprehensive discovery of structural variants. So I'll talk to you in detail about structural variants a little bit tomorrow and a lot on Thursday. But just so that we're on the same page, a structural variant is a large DNA sequence alteration. We typically say over 50 base pairs. And So these genomic rearrangements are often larger than the length of a short read. So it can be complicated to discover structural variants using short reads. We can still do an okay job, and I'll talk to you about the methods that we use for that on Thursday. But we're just able to get a much more complete picture of the structural variation using long reads. So we can see in the top figure that from short reads, we can typically find about 10,000 SVs per genome, but from long reads, we can find about 25,000 SVs per genome, so about 2.5 times more. And the bottom figure is showing what are the specific structural variant types and sizes that each technology is, what are we able to discover with each technology? So it's a little hard to see, but it is showing the number of SVs that are discovered by each technology, with PacBio in blue and Illumina in red. And along the x-axis, the right side is insertions and the left side is deletions. And we can see that the red line is below the blue line. So we're not able to discover as many structural variants with the Illumina short read data as we are with the PacBio long read data. And in particular, we discover a smaller proportion of insertions than deletions. And that's because it's a little bit easier to find where we're missing portions of the reference genome compared to when novel sequence is inserted. It's a little easier to find that deletion signal in short reads. So this is kind of a real life example of the type of information that we're able to access with long reads that we couldn't with short reads. So in 1996, there was a paper that was published on a disease called spinocerebellar ataxia type 4, a very rare, severe disorder. And it was localized to this region on chromosome 16 called 16q22.1, but they were not able to find the exact causal mutation for this disease. They could just tell that it was in this particular region of chromosome 16. And that's all we could really tell for 25 years. It turns out that 16q22.1 is a very repetitive region of the genome. It is very GC rich. It contains segmental duplications, so portions of the reference genome that have been duplicated. It contains pseudogenes, so it's not a very unique sequence of DNA. It's very repetitive, and that makes it basically inaccessible to short read sequencing. But 25 years after this paper was first published, so earlier this year in 2023, they were able to use long read sequencing to identify that the causal mutation for SCA4 was a GGC repeat expansion in this region of chromosome 16. So I think Aaron was talking a little bit about short tandem repeats earlier, or STRs, where there are these units of a short sequence of DNA that is repeated over and over. So in this case, in a healthy individual at this locus, they will have somewhere around 20 repeats of this. It's a GGC unit. So it'll go GGC, GGC 20 times. But in people who are affected by spinocerebellar ataxia type 4, they actually have more like 40 or 60 or 70 repeats of that GGC unit. So you can imagine that with short reads, that all kind of looks the same because the one short read is about 150 bases, would not even span full length of that short tandem repeat sequence. But with long reads, the reads are long enough to span the entire repeat, even when it's expanded in cases of disease, and be anchored to unique sequence on either side of the repeat. And so we can actually get the full picture of how many of these GGC units exist and identify that that's the cause of disease. So this will come up again throughout this session, but I want to highlight that long reads are particularly useful for accessing repetitive portions of the genome for this exact reason. Okay, so to kind of summarize that a little bit, our short reads are about 150 bases long. So if the sequence in the reference genome is repetitive, they may not be able to align to the right place, or the alignment might be ambiguous. So we really can't learn much about those repetitive areas of the genome using short reads. But long reads are usually around 20 kilobases long, so 20,000 bases long. Although that can range from about 10,000 to 100,000 bases long. So they're able to fully span repetitive regions and be anchored to unique sequence on either side. And finally, we can actually sequence and detect variation in those repetitive regions. So this is kind of a metaphor to get that point across. With short read sequencing, you have all of these very small fragments. You can think of them as puzzle pieces, and you're trying to assemble the puzzle to get a full picture of your sample's genome. But this is a very difficult advanced puzzle. And because the pieces are so small, a lot of them look pretty much the same. So it's actually pretty ambiguous where they go in the puzzle. But with long reads, it's like suddenly all of the puzzle pieces are larger. Each of them contains a much bigger proportion of the image, and so it's much easier to tell where they fit. So I said earlier that we can more comprehensively and easily detect structural variants with long reads compared to short reads. So let's kind of take a look at why that is. So on the left, we have an example of a deletion. And on top, we see the short reads from Illumina, and the portion that is deleted completely lacks coverage by those short reads because it's too long to be included in a single read. But in the PacBio and Oxford Nanopore data, we can easily find that deletion because those reads extend on either side easily. In the case of the insertion on the right, we can see that maybe there's something going on. There are all these different colors of reads that probably indicates that there are changes in the insert size and orientation of the paired reads. So there is some signal there, but it is much easier to discover that that's actually an insertion and find out exactly how long that insertion is and what is the sequence of that insertion if you're using long reads and they're long enough to contain that full variant. So long reads make it a lot easier to detect a lot of types of structural variants. But I do want to mention that the lack of coverage and the changes in insert size and paired end orientation in short reads are signals that can allow us to detect structural variation from short reads. And I'll talk about that in much more detail on Thursday. So we can still detect some of these structural variants, but it's easier and we might get higher breakpoint precision when we use long reads. And if this is in a repetitive region, then we might completely fail with short reads and only be able to use long reads. So in addition to being able to more comprehensively and accurately detect structural variants, long reads also provide us information about DNA methylation. And methylation is an epigenetic change to our DNA that influences expression. And it is actually possible to detect changes in DNA methylation with short reads, but only if you add a step to your library preparation and use a bisulfite treatment. So not only do you have to perform an extra step when you're preparing your DNA, but that bisulfite treatment also kind of impacts the sequencing data quality. So it's not ideal. But with long reads, we can access that information about methylation natively through the sequencing technology. So it's much easier. Okay, so now I'll go over how these sequencing technologies actually work. So I'll do a brief review of Illumina sequencing so you can compare, and then I'll talk about PacBio and ONT. So as you probably recall from yesterday, with Illumina sequencing, we shear the DNA into short fragments. We add adapters, attach them to a flow cell, and use localized PCR to create clusters for each read. And as new bases are added, those bases are fluorescent, and we can read that fluorescence to determine what base was added and build up the sequence over time. So this is a technology that uses sequencing by synthesis. And PacBio is also sequencing by synthesis, but with a few different changes. So the first change is that the DNA fragments are obviously much longer. That is kind of the point of long-read sequencing. So once we have our longer DNA fragments, we attach a hairpin adapter to either end. So it has like a little open loop on either end. And that's where we attach the DNA polymerase. So as the DNA polymerase moves along the DNA strand, it actually creates this circular molecule of DNA. And as new bases are incorporated, we can read the fluorescence of the fluorescent bases over time. And similar to Illumina, that's how we determine which base was added and build up the sequence over time. But since we are not just looking at fluorescence, but fluorescence over time, this also allows us to access the information about DNA methylation, because it turns out that a 5-methyl cytosine has a longer incorporation time than an unmethylated cytosine. So the fluorescence for a methylated C would last longer than an unmethylated C. So that's how we're able to get the methylation information directly from the PacBio sequencing. Okay, so this is kind of another image where we see again, we add these hairpin adapters and the polymerase, and it creates this circular molecule. But this one is showing that because the molecule is circular, the polymerase actually kind of just keeps going around and around. And the result is that it will sequence the same fragment of DNA over and over. So you get multiple independent readings of the same fragment. And that allows PacBio to do something that they're calling circular consensus sequencing, or CCS. And what that means is they take all readings of the same fragment and they combine them into one consensus read. And so there might be sequencing errors in one pass through the molecule, but they're unlikely to be the exact same errors in every single pass through the molecule. So when they collapse all of those duplicated readings of the same fragment into a single read, that resulting read is very high quality. It has very few errors. So they call that a hi-fi read for high fidelity. Okay, so we've talked about Illumina, we've talked about PacBio, and now let's talk about Oxford Nanopore. And this is a completely different technology. It does not use sequencing by synthesis. Instead, there is a lipid membrane, and there is a voltage that's applied across that membrane. But because it's a lipid membrane, the ions can't pass through that membrane. So the only way that they can pass through is through a very small pore called a nanopore. So a single strand of DNA passes through that nanopore, and as each base passes through the pore, it has a certain characteristic resistance just because of its chemical makeup. So they can read the resulting current and determine the pattern of bases from that current. So we can see in this image that an A has a certain level of current, a C has a higher level, a T has a lower level, and that's how they can determine the sequence. Of course, the reality is always a little bit noisier than that idealized diagram. You can see some real data. It's a little bit messier. So actually, Oxford Nanopore has developed machine learning algorithms to be able to denoise that data and perform their base calling. I want to also mention that the methylated bases have a different current, a different resistivity from the unmethylated bases. So that's how Oxford Nanopore is able to detect DNA methylation during sequencing. Okay, so how long are these long reads actually? For PacBio, we typically see read lengths that are somewhere around 15 to 20 kilobases. They don't go ultra long because they want to be able to read the same fragment of DNA multiple times to get that high fidelity read. Oxford Nanopore is not really limited by their sequencing technology. They could kind of go forever, but it turns out that it's difficult to prepare ultra long DNA fragments just in terms of the library prep. So we typically see about 20 to 30 kilobases from Oxford Nanopore. But it is possible to get reads that are even longer than that. The longest read was over 1 million bases long. Okay, so what are some of the applications of long read technologies, and what are some of the cool projects that are using long reads? One of them is the All of Us research program, which earlier this year released over 1,000 long-read genomes. So for long reads, because it's a newer, more expensive technology, 1,000 is actually quite a few. So this is one of the larger population references of long-read data, and they're hoping to increase that and sequence something like 10 or 20,000 long reads and release those in the coming years. And so that will be a useful population reference data set for this new technology. Another project using long reads is the Rare Genomes Project. So this project is aiming to find causal genetic mutations for very rare diseases. Some of these diseases are so rare that there are not really any other studies that are researching them. So for this patient who had a severe neurodevelopmental disorder, they first did short read sequencing, and they found some sort of signal, this pileup of split reads. So they could tell that there was some sort of structural rearrangement going on there, but they weren't able to fully resolve that event with short reads. So they did long read sequencing, and it turns out that this patient had a chromothripsis event. And chromothripsis has been described as chromosome shattering. So a whole bunch of different chromosomes broke apart and then got stuck back together in ways that they were not supposed to be stuck together. So this is a pretty rare and deleterious type of event. Another patient from the Rare Genomes Project, this one had some brain abnormalities, and they first did whole exome sequencing, and they did CNV calling on whole exomes, and I'll talk about how we do that tomorrow. And they were able to detect that there were 2 regions on chromosome 6 that were duplicated and 1 that was deleted, but it's not a very high resolution method, so they couldn't find the exact breakpoints. They couldn't tell exactly what was going on. So they did long read sequencing, and they were able to find the breakpoints of this event and actually determine that it was a complex structural rearrangement that also included an inversion in addition to the copy number variants that they previously detected. Okay, so we've talked about how the long read sequencing technology works and some of the applications of long reads, but how do you actually process the long read data? So the Broad has created and published several long read pipelines for use processing long read data, but actually these pipelines use open source tools that were developed by other groups outside of the Broad. But they're really good tools, they work well, so we didn't feel the need to reinvent the wheel. We just want to make them easier to run by creating and publishing these automated pipelines. And these pipelines are written to be run in the cloud, so they take advantage of parallelization to process this really large amount of data, but they could also be run on a high performance computing environment. So the first step of the pipeline is to align the reads back to the reference genome. That's familiar from short reads, but for long reads, we use an aligner called Minimap2 because it's a really versatile aligner that also works for long reads. But one note about using Minimap2 is that you have to make sure that you're using a version of the HG38 reference that does not contain alternate or decoy contigs. So actually for short reads, the default reference that we use does contain alternate or decoy contigs that describe diverging haplotypes. And BWA can handle those without impacting mapping quality over those regions. But Minimap2, the best practice is to use a reference without those alternate or decoy contigs. So just make sure you're using the right reference because it will be different from the one that you use for short reads. After alignment, we call short variants, so single nucleotide variants and short insertions and deletions. And the tool that we use for that is deep variant, which is a variant color that's based on a convolutional neural network. And then, of course, we're very interested in structural variants because we're talking about long reads. So we call structural variants, and the team at Broad has tried several different tools for this, including PBSV and Sniffles2. And I won't go through the algorithm for this in detail, but I do want to point out that the first step is to find SV signatures by looking in the cigar string for deletion and insertion signals of greater than 50 base pairs. So they can actually go directly to the cigar string to find evidence for structural variants as the first step of the algorithm. So just remember that when we talk on Thursday about calling structural variants in short reads, where it's a lot more complicated than that. A final optional step is to perform a de novo assembly of the sample genome using a tool called Hi-Fi ASM. So with short reads, it's really difficult to perform a de novo assembly of a genome. It took a lot of effort to build the reference genomes over the years. But with long reads, we're able to construct much longer, more accurate haplotypes and actually perform whole genome assembly for a given sample. And so that's kind of a new and exciting way of exploring genome variation that's become possible with long reads. All right, and that's all I have on long reads. Any questions? Yes.
Please turn to the page 3.
Okay.
Yeah. I would like to ask the picture bottom left. Because I'm interested in bio-nano. There's a lot of very large structure variation. So what are they? Because what are they? The bio-nano detection over the 10 kb.
Yeah, I have to admit I don't know a lot about bio-nano, so I don't know the answer to that question. All right, Eric's saying he doesn't really know either. I'd be happy to look into it later and try to get back to you.
Thank you.
You can also see that this figure is from this paper Chason et al. 2019. So you can look for that paper. That's where that figure is from, and I bet they have an explanation. Yeah. All right. Other questions? I'll just give it a second in case anyone from the other room ends up having a question. Okay, I'm not hearing any other questions. So that's actually our last session of the day. So we'll end here for today, but the instructors will stick around for a little while to answer more questions if you have them. All right, thanks everyone. Oh, we do have one question. Sorry about that. Ended a little early. Ah. Yeah, so this question is referring to the figure in the top left of this slide, where we can see that there are actually some structural variants that are only called in the short read data and not called in the long read data. So, you know, the question is why? The long reads are supposed to be really good at this, so why are we missing some things? That turns out to be a little bit of a complicated question, and there's a really good paper on it that I can share with you right after this. But one main factor is that as you get to a certain length of structural variant, it's about like 5 to 10 kb, they start to maybe not be fully contained by the long reads, or like harder to call from the long reads because maybe the long read didn't span the entire variant. And so those structural variants are a little harder to detect with long reads just because of their large size. But with short reads, we can actually detect them using changes in read depth. So that is one class of structural variation that's currently better detected by short reads than by long reads. But if you want to come up here afterwards, I can try to find that paper that talks about this in more detail. Okay, I think that's it on questions. So
Thank you, everyone. We'll see you tomorrow.
yeah, thanks everyone. See you tomorrow.